In this video, we're going to discuss the DNA, RNA, or in the series of videos, we're going to talk about DNA, RNA, and protein-based lab techniques. The goal is to introduce you to the technology, even though at this point in the course, some of it may not make total sense. We will come back to many of these techniques throughout different modules, and I'm also going to link some helpful videos that you can watch if these methods don't make sense. You can also ask me, as I have done most of these methods countless times, usually incorrectly to begin with. So if they don't make sense, please send me an email. We'll set up a WebEx. I can just respond to your email. We'll go from there. But let's start with DNA-based technology. So the most widely used is the polymerase chain reaction, or PCR. You use PCR in the lab when you want to create lots of copies of a piece of DNA, say from a crime scene or in a COVID test. PCR uses a temperature-stable polymerase. Remember, polymerase is the thing that copies the DNA. And this temperature-stable one is from bacteria. It's called Taq polymerase. So to do a PCR, you're going to add a small amount of DNA, your temperature-stable polymerase Taq, some excess nucleotides, and primers. Polymerase, as we're going to talk about later, needs a primer to first bind to the DNA before it can land and then replicate the DNA. So what you do is you're going to cycle through different temperatures, heating it, which will separate the helix, lowering it a little bit so that the primers can bind or anneal, and then raising it to the ideal temperature for that Taq polymerase, which is around 72 degrees Celsius. When you do this over and over and over again, like 30, 35 times, you're going to get an exponential increase in the amount of DNA. So this is shown here. We have our first denaturation with heat, and then we cool it to anneal, and then we extend it at 72 degrees. And if we cycle through the first cycle, we're going to go from one piece of DNA to two, 2 to 4, and this will increase exponentially with each cycle. We also have gel electrophoresis. So when you PCR a sample, you want to see that it works, that you ended up with a lot of DNA. So what you're going to do is you're going to run your sample on a gel. And gel electrophoresis separates things by size, by weight. So for DNA, you're going to make your gel, and then you're going to add to the first well here a molecular weight standard, which is going to give a line at a known weight when you can look that up. Usually it comes with the package, just like a little piece of paper. Then you're going to add your DNA samples that you've mixed with a loading dye so you can see it. It's usually blue. And when you apply a current to that gel, the negatively charged DNA is going to move towards the positive electrode. And the short fragments are going to move faster because they're able to bob and weave and squeeze through those gel particles and get to that positive electrode at a faster rate. You can then stain your gel with a dye that binds to nucleic acids, and it gives you these fun little lines here, these bands, and you can compare it to the ladder to say, oh, this is a 100 base pair band, and this is a 200 base pair band. So say your DNA piece that you want to copy is really, really big, or you want to change the sequence around, you want to genetically modify your piece of DNA. This uses recombinant DNA technology, or when you have mixed DNA from two sources. To get recombinant DNA, you're going to take advantage of a class of enzymes from bacteria called restriction enzymes. These cut the DNA at a specific site called a recognition site. So if you cut your DNA with your enzyme and you cut your vector with the same enzyme, you're going to have complementary overlaps. So the overlaps are shown here. If you cut with EcoR1, for example, you're going to get this overlap where the sequences are complementary. They're going to want to come back together. So if you are making that plasmid, you do the same thing. You take your plasmid, you cut it with a restriction site, you take your DNA of interest, you cut it with that same restriction site. They should come together. They should want to join together. And you're going to join them with an enzyme called ligase. You can then put your vector, your circular piece of DNA, into bacteria, and they're going to make millions and millions of copies of that vector for you. Now, the trick is figuring out which bacteria, one, have taken up your plasmid, and two, which of those plasmid vectors you've actually put your foreign DNA in. And so you're going to use a double system here for selection. The first is that the plasmid or the vector itself is going to have a gene that gives the bacteria antibiotic resistance. So if you introduce an antibiotic, all the other bacteria are going to die. Usually we use something like ampicillin. So if your bacteria has taken up that circular piece of DNA, it can withstand the ampicillin. If it hasn't, it'll die. So that's the way that we see that we have that vector, that plasmid in our bacteria. Now, to check that it's cut, you can do something called blue-white selection. So if you grow your bacteria on an X-gal plate, a bacteria that has a LAC-Z gene are going to eat the X-gal and turn bright blue. However, if you make your restriction site, if you put your DNA of interest in the middle of that LAC-Z, you've messed it up. So now they can't turn blue, and instead they'll be white. And then you just pick the white colonies that show your broken LAC-Z gene. Finally, let's talk about Sanger sequencing. This is a way to determine a DNA sequence, and it uses modified nucleotides. The modified nucleotides have a fluorophore on one end so that they're colored, with each letter being a color. So A would be red, G's would be yellow, C's would be blue, and T's would be green, for example. These modified nucleotides are also altered so that when they're added to a growing strand of DNA, they're going to stop the addition of more nucleotides. They don't allow for that bond to form with another nucleotide, and so the sequence will just end. They'll always end in a color. And so what you can do is take your sample of DNA and take your polymerase and take mostly normal nucleotides, but a few of these modified fluorescent nucleotides, and mix them all together, and then separate them in a version of gel electrophoresis that goes through a small capillary, and it has a camera. And so the smallest pieces are going to go first. And so say the smallest piece right here is shown as a TTGTC, and the C is modified and it's blue. So the first thing that they'll detect is blue. The next biggest piece would be the addition of one more nucleotide, and that in this case is a G, which is green. And so it will read blue-green, and you'll know that the sequence then, or the computer will tell you, the sequence then is a C and then a G. And it does this all the way through, getting larger and larger fragments, and from that you can tell the sequence of the nucleotides. So in this video, we talked about some key DNA technologies, and next time we will talk about RNA technologies.